Our second lecture in molecular pharmacology is going to be on transcription. For this, you need to be able to describe RNA, its components, structure of RNA, and the different types of RNA. You should also be able to describe the process of RNA transcription, the enzymes involved, and also how transcription can be a pharmacological target. So first of all, I'd like to highlight the differences between RNA and DNA. I suppose the major difference between them is that RNA is a single-stranded molecule, as indicated in the diagram on the right. It's a single helix, whereas DNA is a double helix, a double-stranded molecule. The second major difference is in the nitrogenous bases. So while both of them have 4 bases, the types of bases are slightly different. So DNA has A, G, C, and T, adenine, guanine, cytosine, thymine, whereas RNA has A, G, C, and U, where U stands for uracil. And this is probably one of the major differences. The second major difference in the components is the sugar itself. So the sugar in this case is ribose, whereas for DNA it was deoxyribose, and this is highlighted on the bottom left diagram at the 2' carbon, or the second carbon. And the second carbon in the case of DNA has no oxygen attached to it, whereas the second carbon in the case of RNA or ribose has an O attached to it, so an OH molecule is there instead of a H, and there's the major differences. So while a lot of people will have heard of messenger RNA, this really only comprises about 3 to 5% of the total amount of cellular RNA. Most RNA, in fact, is actually ribosomal RNA and is present as a basic structure at the ribosomes. In addition to that, there's also transfer RNA, which is involved in protein translation, and we'll talk about in the third lecture. And finally, microRNAs or other non-coding RNAs, which have been discovered more recently, and these are involved in the regulation of gene expression. So just briefly, the diagrams shown on the right and the bottom left highlight some of these different forms of RNA. At the top right, we have the ribosomal RNA, which, again, as I said to you, we'll discuss when we come to talk about translation. And the second one down shows what we're going to be talking about today, which is transcription and messenger RNA molecule. The transfer RNA molecule is in the bottom right, and then finally the microRNA is on the bottom left hand side. So transcription involves the unwinding of DNA to expose bases on each strand, one of which acts as a template for RNA synthesis. So it's quite similar in that respect to DNA replication and requires the unwinding and opening up of the double helix. And one of these strands acts as a template for RNA synthesis, meaning that one of them is only read and used as the template. So when new RNA is being synthesized, it does not hydrogen bond to the template. This is where it, I suppose, is different from DNA replication. Whereas in DNA replication, the new nucleotides were added on to the old strand. In this case, the new RNA does not hydrogen bond to the template. This allows for the immediate release of the new mRNA molecule, and also means that many RNA copies can be made of the same gene in a relatively short time. You're not relying on the bonding and then removal of those bonds if you need to make more copies of an mRNA molecule. Most mRNAs as well are no more than a few thousand nucleotides in length. So transcription is catalyzed by a different enzyme to DNA replication. Rather than DNA polymerase, it's RNA polymerase, and again involves the formation of phosphodiester bonds that link the nucleotides together in a linear chain. But this linear chain, just to reinforce the point, is not attached to the DNA molecule. So the growing RNA chain extends in a 5' 3' direction, meaning that it goes from the 5' carbon position on the deoxyribose to the 3' direction, with ribonucleotides ATP, CTP, GTP, and UTP added on to each other. And RNA, unlike DNA polymerases, can start without a primer. So transcription is not as accurate as replication. RNA does not permanently store genetic information, so it's not like DNA, where DNA is copied and every single cell in the body has the same copy of DNA, or at least in theory they do. RNA can vary ever so slightly. And RNA polymerases make mistakes 1 in every 10 to the power of 7 nucleotides. So 1 in every 10 million nucleotides that are synthesized by RNA polymerase is actually a mistake. But they can perform excision reactions similar to that of DNA polymerase, as mentioned in the first lecture. So we're going to be mentioning three different RNA polymerases in particular. Each of these polymerases have 2 small and 2 large subunits. These combine together and give, I suppose, what the diagram on the right kind of looks like. So the 3 major types are RNA Pol1, Pol2 and Pol3. RNA polymerase 1 synthesizes ribosomal RNA. RNA polymerase 2, which we'll be focusing on today, synthesizes messenger RNA and also some other nuclear RNases. And RNA polymerase 3 synthesizes transfer RNA, which we'll come to talk about in the third lecture. So there are 3 phases in transcription: the start, middle and end, or initiation, elongation and termination. And these are indicated by the diagram on the slide here. So as mentioned already, DNA is a double-stranded molecule. In order to get at the nucleotides to read them, the enzymes need to unzip the DNA, they need to unwind it, and they need to get the RNA polymerase enzyme, in this case, to bind to the DNA in order to read it. And in order for the RNA polymerase to do that, it needs something that it recognizes, and this is known as a promoter region. So genes that are turned into protein will very commonly have what's called a promoter region, which is a small region of DNA that has the same or similar nucleotide sequence and is able to be recognized by its 3-dimensional shape and structure by the RNA polymerase enzyme. And the RNA polymerase enzyme binds to the DNA and starts to read the nucleotides that are present on the DNA, the A's, C's, G's and T's. In parallel to that, it starts to synthesize the new strand of mRNA. So point 1 is that RNA polymerase binds to the promoter and starts to unwind the DNA as it moves along. The RNA polymerase reads the DNA template from the 3' end to the 5' end and produces the new RNA or mRNA molecule, or transcript as it's called, by adding nucleotides to the 3' end. So it's synthesizing it in a 5' to 3' direction as indicated by the diagram. So in part B there, the elongation, you can see that the direction of transcription is in the 5' to 3' direction, but it's actually reading the DNA strand in a 3' to 5' direction. Each time it's taking new nucleotides or nucleoside triphosphates, A, U, C's and G's, and it's adding it on to a growing strand. You'll notice as well that as the RNA polymerase moves along, that the green mRNA molecule is not physically bonded to the DNA. This is what I was referring to earlier, meaning that it doesn't hydrogen bond to the DNA, it actually detaches. And this allows the RNA polymerase to go back over the same gene again several times and almost photocopy it. So it's not hydrogen bonding, meaning it can physically move away from the site of the DNA and travel to the ribosome to get translated into a protein. Finally, the termination occurs when the RNA polymerase reaches the termination site. The RNA transcript is set free from the template and you have a new mRNA molecule. You'll notice as well that as the RNA polymerase is moving along, it's obviously unzipping the DNA as it's moving along, but as it goes past the different DNA regions, the DNA itself is winding up again and hydrogen bonding again with the complementary base pairing method. So it closes up as soon as it's been read. So for a gene to be read and to be transcribed, there needs to be a lot of control built in, because you can't just have DNA being read and transcribed all of the time. Otherwise, we'll be making too many mRNA transcripts and we wouldn't be able to control the amount. So control elements are built into our genome and the genomes of many other animals. And these control elements may be near or they may be very far away from the start of the gene or the promoter sequence. And this is indicated in the diagram on the right here, where you have control elements known as enhancer sequences. You have promoter sequences, which you've mentioned already, and that's the area where the RNA polymerase enzyme first binds. And then as indicated by the arrow and the region in orange, you have the transcription start site. So this is where it begins to synthesize new mRNA molecules. So promoter elements are known as proximal elements, and they're usually within 200 base pairs of the start site and are usually around 20 base pairs in sequence length. Enhancers, on the other hand, can be a long distance away, perhaps even up to 10,000 base pairs away from the start site, up or downstream. And this may seem strange in thinking that you could have something so far away that could have an effect on it. However, if you think about the fact that DNA is highly folded, and as shown by the diagram there, you can see the folding of the DNA. Something that is a sequence of DNA that is far away, like an enhancer element, can in reality be quite near, because with the folding of the DNA, it can end up being literally right beside it, as shown in the diagram on the bottom right. And this can actually have an effect and enhance the transcription of the DNA. So transcription factors stimulate or repress RNA transcription. So it's not just good enough to have the RNA polymerase bind to the promoter sequence. This needs to be regulated, and this requires the use of proteins known as transcription factors. So whereas previously we were talking about control elements, these were in cis or in sequence with the DNA. It was a piece of DNA. These trans-activating factors are often proteins, and they are required to bind to promoter regions in order to allow for transcription to occur. So transcription factors can stimulate or repress transcription. The transcription factors bind to the promoter elements and sometimes as well enhancers, and these recognize specific sequences of DNA. So they're able to recognize it, they're able to bind to the DNA and either enhance or repress transcription depending on the signal that it's receiving. So in order to start the process off, promoters will, as we said already, will determine the site of initiation and direct the transcription factor binding. And there are three types of promoters that have been identified in eukaryotes. The most common of which is the TATA box, or TATA, thymine, adenine, thymine, adenine. So once this sequence is found near the start of a transcription site, it can act as a promoter sequence. And this is shown in the diagram on the right, whereby a TATA sequence is near the start of the gene. It is recognized by transcription factors, which then can bind the TATA box and start off the whole process, acting as a scaffold for other proteins to join in the process. So DNA is double-stranded, so in principle 2 different RNA molecules could be transcribed from any gene. Promoter DNA sequences are asymmetric, meaning that if you have a TATA on one side, you have an AT on the other side, because the complementary binding process. So promoter DNA sequences are asymmetric, but RNA polymerase only recognizes one sequence and will therefore bind in only one orientation. So transcription factors listed here, transcription factor 2A, 2B, 2D, etc., are specific for certain RNA polymerase. In this case, because it has the 2 after it, it's specific for RNA polymerase 2. So these transcription factors act as a structure in which the RNA polymerase can bind to. So transcription factor 2D contains a TATA box binding protein, or TBP. So it's this transcription factor that can recognize the TATA box, and then a TATA box binding protein recognizes that particular protein. This allows then the assembly of a multi-protein complex, and up to 12 other proteins known as TATA box binding protein associated factors, or TAFs, can join in. So this is like a Lego house, essentially, that you have one protein binding to the DNA, another protein recognizing that protein, etc., etc., and they all add on to each other in sequence. This is known as a transcription initiation complex, and this is very, very necessary in order to control the whole process, because if RNA polymerase was just able to bind to DNA, it could transcribe any gene at any time, and you mightn't have the requirement for those genes to be transcribed. So there's a high level of control built into this. In addition to that, the C-terminal domain of RNA polymerase is phosphorylated during the initiation of transcription, and this is necessary for the whole process to kick off. So RNA polymerase 2 gains access to DNA by the DNA helicase, and we've said that already. This is necessary to unwind the DNA. It also requires chromatin remodeling enzymes. So, as we've said already, the DNA is wound up very, very tightly around these proteins known as histones in the nucleosome complex, and in order to unwind that and to get the DNA accessible to the RNA polymerase, you need to unwind the DNA, but you also need to remove these histone proteins, and that's where the chromatin remodeling enzymes come in. As you begin the DNA process and start to add on the nucleotides of mRNA, the elongation process also needs to be controlled, and this elongation is associated with what are known as elongation factors, an example of which is called elongan, and this is shown in the diagram on the top right. So as the RNA polymerase is moving along, it can be quite difficult for it to stay on track or to read the DNA correctly. So it requires these elongation factors to guide the RNA polymerase enzyme along the DNA to make sure that it stays on track and doesn't detach. And the proteins also decrease the likelihood of dissociation or unbonding of the RNA polymerase molecule. And these typically will associate after initiation. So once the transcription initiation complex has been formed, once the RNA polymerase starts to move along and read the DNA, these elongation factors will bind and guide the RNA polymerase along the sequence. So transcription will stop at varying distances downstream depending on certain signals. So as we've been saying already, there are more than just a couple of proteins required to begin the RNA transcription process and also to stop it. So we have proteins that positively promote RNA transcription, ones that stop it. These are known as activators in the case of promoting it, and repressors in the case of stopping it. So activators are proteins that bind to genes at sites known as enhancers, and they speed up the rate of transcription. They have a DNA binding domain, meaning that they recognize the nucleotides on DNA, and can have a linker region as well, which is able then to link the protein itself to the transcription initiation complex. On the opposite side of that, then you have repressor molecules, and repressor molecules are proteins as well. They can also recognize DNA, but rather than binding to enhancer regions of DNA, they bind to silencer regions, and these slow down or stop in some cases the transcription process when it might not be necessary. So our diagram on the right here shows that, and it shows how activator proteins can bind to enhancers and inform the activation of the transcription initiation complex. In contrast to that, then we can also have the silencers, where the repressor molecules will interact with, and they can stop the physical moving of the RNA polymerase molecule along the DNA coding region or the gene. So the activity of these activators and repressors is really important, and changes can occur in chromatin structure because of these particular proteins. So these may be necessary during different phases of cell activity in order to transcribe certain genes, in order to stop the transcription of certain genes. If you have a physiological response, for example, if you need to make hormones, if you need to make cytokines for immune responses, you need to very, very quickly be able to turn on the DNA for gene transcription. But equally then, when the response is over, you need to be turning it off. And this is where these activators and repressor proteins actually come in. So they have a direct effect on the assembly of initiation complexes, but then equally they're able to stop it once the physiological response is no longer needed. When DNA is open and responsive to initiation of transcription, it's called euchromatin. In the cases where it's condensed and closed up and relatively inaccessible, it's called heterochromatin. So once the mRNA molecule is completed in its mature form, it has a number of noticeable features, and these are highlighted in the diagram on the right. We have what's known as a 5' cap, which is on the 5' carbon end, as shown on the left-hand side here. We have a 5' untranslated region, which, as the name suggests, doesn't get translated. We have a translated or coding region indicated by the section in green in the diagram, and this is what eventually will go on to make the protein. We have a 3' untranslated region, which again doesn't get translated at the ribosomes, and then we have a poly-A tail. So this molecule, as we know, is created near the DNA inside the nucleus, but it needs to make its way from the nucleus to the ribosome, which is out in the cytoplasm. In order to do this, it needs to obviously have certain signals attached to it in order for it to be transported, but also to stop it from being degraded. Our cells contain enzymes called nucleases, which are relatively quite active due to the chances of being infected by viruses, which of course contain DNA and RNA sequences, which we normally will try and break up and stop them from replicating. So we have this natural defense mechanism to degrade nucleotides, but we want the mature mRNA to make its way from the nucleus out to the cytoplasm, to the ribosomes. So the 5' cap is one such signal that protects it, and the poly-A tail as well is a signal that's used to export it from the nucleus into the cytoplasm and eventually make its way to the ribosome. So this capping process occurs at the end of the chain, as we've seen at the 5' end, and it involves the addition of 7-methylguanosine to the 5' end. And this is essentially used during the initiation of translation. And this is what it happens at the ribosome, as we'll see in the third lecture. So it requires this 5' cap in order to begin the translation process of mRNA. The 3' polyadenylation signal, or poly-A, can result in the addition of up to 250 A residues or adenines at the end of the mRNA molecule. And this is important for nuclear transport and also for mRNA stability, so that it can make its way from the nucleus to the cytoplasm. So when it comes to RNA being transcribed, the way in which RNAs are read and transcribed will be determined by the genome, and the genome and how it's organised has only really been discovered relatively recently. So not all genes not all pieces of DNA are genes as such, and not all genes then are coded for into protein. So most genes are actually discontinuous, meaning that they're not all read in the same sequence or in a continuous sequence. There are those in particular in eukaryotic organisms. So there are those regions or sections that are coding. These are called exons, and those that are non-coding, which are known as introns. And the number and size of introns varies with different genes. You can see in the diagram at the very, very bottom, this is the human factor VIII gene, which codes for the protein factor VIII involved in the clotting process. And you can see that the coding region is up to 200,000 nucleotide pairs and made up of 26 different exons. But there are large sections of it which are non-coding, and these are known as introns. These introns are removed by a process called splicing. So while the mRNA is synthesized, not all of it is actually used, and it needs to be removed by splicing. And these are indicated by certain splice sites or sequences that indicate splicing. In addition to that, what is useful about this, having the genes in the discontinuous fashion, is that you can combine different exons together to form different proteins. So you don't always have to combine all of the exons together in order to form protein. You can combine different variations of these exons. So, for example, if you have 26 exons, like is indicated here in the diagram at the bottom, you could maybe combine 10 of those together to form a protein, or 26 of them to form a protein, and in each case you end up forming a different a different final mRNA molecule, which then will lead to a different protein being formed. So from the same genetic information, you can end up getting a multitude of different proteins rather than just the same protein every single time. And this is called alternative splicing. This does actually occur for certain genes, particularly you get tissue-specific splicing, whereby one organ, for example the brain, might have one version of a gene being spliced, and in the liver, for example, you get a different gene being spliced and turned into a different form of the protein, or splice variant as it's called. Since the human genome was decoded many years ago, a lot of work has taken place on the sequencing and the annotation of these sequences. It's been discovered though that a very small percentage of our genome is actually protein coding. Quite a large amount of it is introns and other sequences, in particular leftovers from infections from our ancestors, so pieces of DNA and retrotransposons, for example, that have incorporated themselves following infection into our DNA and then got passed on to successive generations. Other genes have been duplicated. Other sequences are repeats. We saw the example already of telomeres in lecture 1. There are other sequences like that are repeat sequences that are necessary for certain processes within the nucleus. So finally, I want to mention the targeting of RNA transcription by drugs in order to either induce a positive effect or a negative effect. The first of these that I want to mention are poisons. So there's been a number of deadly poisons and toxins that have been identified that can interfere with RNA transcription. Of course, RNA transcription is essential for life. We need to transcribe our RNA in order to make protein and for our cells to survive. So if you have a toxin or a poison that can target this enzyme, it can be deadly. In particular, I've mentioned here the poison alpha-amanitin, which is comes from the death cap mushroom, Amanita phalloides, and the target of this particular poison is the RNA polymerase 2, so the enzyme that's involved in mRNA transcription. And the consequence of it binding, which is it's only a small 8 amino acid cyclic peptide, is that it inhibits polymerase activity and in particular causes liver failure and death. The second case where it could be used positively is as an antibiotic. So the example I've given here is Actinomycin D, and it's derived from soil bacteria, and it can bind DNA and prevent elongation by RNA polymerase. So this means that bacterial RNA polymerases can't elongate, can't transcribe their RNA, and can't make protein, so they eventually die. So this can be used as well in the case of cancer chemotherapy in some cases, and prevents the transcription of RNA. So in summary, RNA is different from DNA. It's a single helix and has different forms, different cellular RNases. Its purpose, the purpose of transcription is to make messenger RNA, and transcription can be a drug target. It has antibacterial and also poisonous effects.